Well, thanks very much, and thank you especially to people from different departments who have come here together. I think it speaks volumes for plant biology on campus that we're in different places on campus. We're in different departments, sometimes different faculties, but we have some common interests, and I think if we can have more venues like this, we can demonstrate to the powers that be, particularly this year, that plant biology in general and forest trees in particular are a going concern on this campus. I also want to say thanks to those who have come, had to pack a lunch to make the great trip across the river for the presentation day. It's greatly appreciated. So as Frances said, last time that I gave a presentation here, I talked about spruce dormancy, and I was young, pregnant, and untenured. So now I'm tenured. I'm not pregnant. But I'm going to tell you about a completely different project that's taken place largely in the intervening time, and that's on mountain pine beetle. So I'm going to start with an overview of the outbreak. This is nothing new to most of you in the room. Most of you are well familiar with mountain pine beetle because you work on it or because you've heard seminars on it. So this will be really brief because I want to get to two parts of the TRIA project that I'm involved in. One in collaboration with Dave Coltman, whose work I'm going to talk about today in conjunction with Kathy Cullingham on the population genetics. And another part where we're doing more physiological, biochemical, and genomic analyses, looking at what the differences and the similarities are between these two hosts for mountain pine beetle, and trying to figure out what the effects of abiotic stresses are on those responses, and then finally finish with a few perspectives. So as is typical, when you finally get a chance to talk for 50 minutes, you really want to tell the whole story, and I realized at noon that I have a lot of slides in here. Because I'm really excited to tell you about the full story and some new data that are just coming out of the lab. So I'm going to talk really fast, and I apologize for that. But I do want to leave time for questions at the end. So today I'm going to talk about the work that two people have really largely led since the onset of the project, Kathy Cullingham, Adriana Orango. Adriana's here today. And I also want to talk about some work that Jill has been doing in collaboration with Adriana. So these three people are largely responsible for all of the data. So all the cool stuff that I'm going to show you is what they've done. If there are any mistakes, that's me. So I know you all are familiar with mountain pine beetle, or the vast majority of you are familiar with mountain pine beetle. We've all seen beautiful pictures or lousy pictures, depending on your perspective, of large tracts of the landscape that have been turned red by attack by this beastie. I don't want them too low. It is post-lunch. Which, when you do get down onto the ground, we see these red trees, or sometimes you don't see them. But after a few years, this is what you see. And this is actually just down the road from where I grew up in Prince George, where a bulk of the devastation has taken place. And so these grayed out trees in the landscape are a stark reminder of the ecosystem change that can be brought about by something that is often compared to a grain of rice. So mountain pine beetle can make that kind of devastation because they mass attack their hosts. And so if sufficient numbers of mountain pine beetle get together and attack a pine tree, they can overcome the defenses, such as this pitch. This is an induced response that's brought about by the attack on this tree. And if there are enough beetles, they overcome this, they can successfully colonize the tree, create galleries like this, offspring result, and those offspring fly the next year to colonize more trees. They don't do it alone. There's a whole community that hitchhikes on these beetles, including these fungi. There are multiple different fungal species, fungal associates of mountain pine beetle. And these guys colonize the xylem and the phloem, so the wood and the bark. And not only do they make this more palatable for the beetle, extracting nutrients that then the beetle gets through eating the fungus. So they're making the hotel more palatable. But would you believe that these fungi actually can take those defenses of the beetle, they can detoxify them and use them as a carbon source to further fuel not only the beetle's ability to colonize this tree, but provide it with nutrition from the very defenses that the beetle has invoked. So, I mean, how insulting to the tree. So, mountain pine beetle, historically, within the range of time that we have measured mountain pine beetle outbreaks, not me personally, I haven't been around this long, has been really, in Canada, focused on the central part of British Columbia. This is the first half of the current outbreak, which started over here in central British Columbia, fanned out from there, following some long-distance flights in 2006, which we've documented genetically, which others have documented using a lot of diverse measures, including holding butterfly nets, so windows of airplanes and crazy things like that. The beetle ended up on the other side of the Rockies in the northern part of Alberta and has subsequently migrated from there, including into this area, the dark green here being lodgepole pine, the light green being jack pine, and the overlap of those two ranges being where jack pine and lodgepole pine, which are considered sister species phylogenetically, hybridize. And so Francis Yeh has spent a great deal of his professional career looking at this zone of hybridization. But with the tools of the day, it was still difficult to really define this zone of hybridization. So one of the things that we set out to do when mountain pine beetle first arrived in northern Alberta was to figure out what was that zone of hybridization, and could we really distinguish lodgepole from jack pine and their hybrids, because morphologically that's not simple to do. So we set out to do that with molecular markers. So Kathy developed a series of microsatellite markers and then later SNP markers, which are single nucleotide markers that we can interrogate on a much larger scale and more economically than we can with microsatellite markers and more conventional markers, looking at several populations over the zone of lodgepole pine, jack pine, and this putative zone of overlap. And so based on her analyses, she could bin the individuals either as jack pine as blue, lodgepole pine as red, lodgepole as gold, or hybrid as red. So using this information then, she developed this interpolated surface to describe the zone of hybridization between jack pine and lodgepole pine. And so this is overlaid actually on new data that has come from the federal government on pine distribution. And so you can see that that pine distribution is different than the classic little range, as is the size of the zone of hybridization. So Kathy's done some other analyses on the hybrids, showing that they have greater lodgepole pine ancestry than jack pine. So within the hybrids, there's a greater contribution of lodgepole pine than jack pine genomes. She's also gone on to look at the larger population structure, and this is the first part of that analysis, that shows that we can distinguish, like other groups, a northern and a southern population of lodgepole pine in the region that we sampled. But differently than other groups, we can distinguish two populations of jack pine from central Canada and western Canada. So that's different. And the reason why we think this is different is because we used a lot more markers than had been used in the past. And so what we see then is that part of this genetic structure is due to intergressing genes. So lodgepole pine genes intergressing into jack pine intergressing into lodgepole pine. So at least part of this population structure is due to the integration of the other genes into the other species. And so that we see now that this zone of integration is much larger than many of us would have ever predicted early on. So Kathy, along with another postdoc that was with TRIA, Patrick James, did some species distribution modeling. I don't do math in public, but they do, fortunately. And so what they're able to show is that we can describe the location of jack pine and lodgepole pine as a probability based on environmental variables, such as elevation. The higher up you go, the less probability you have of finding jack pine. The drier or the less moisture availability that we have on the landscape, the more likely we are to find jack pine. Low temperatures, more likely to find jack pine. And then, of course, their distribution on the landscape. So we can predict, using geography and habitat, where we would find lodgepole and jack pine. And that's allowed us to come up with these species distribution maps and hybrid zone range. So what she was also able to do is use those markers to interrogate jack pine, lodgepole pine, and hybrid that have been attacked by mountain pine beetle to show more objectively that pure jack pine had been attacked by mountain pine beetle as early as 2010. So this was really important. There was a long-standing debate at that point of whether mountain pine beetle would be able to effectively move across this hybrid zone, whether they would effectively colonize natural stands of jack pine. One of those reasons is because this bridge, as it's called, has a fairly low distribution of pines. But there was this question about whether jack pine would be a suitable host. And it really mattered to provinces east of Alberta, because certainly in Saskatchewan, when the question about mountain pine beetle came up, and the chief entomologist there asked for funds to get ready for mountain pine beetle control, his minister said, well, let me know what's in jack pine. Until then, it's somebody else's problem. So being able to demonstrate that mountain pine beetle had moved to jack pine enabled spread control to become a reality for the province of Saskatchewan, has enabled the provinces of Manitoba and Ontario to put in place mountain pine beetle action plans in the event that mountain pine beetle moves there naturally or is accidentally introduced, say, by firewood or these sorts of things. So this finding was important on a federal policy level and on a provincial policy level. So today, I should actually be asking Anina, who probably knows this better than I do. We know that mountain pine beetle has crossed the 60th parallel. We've got this famous picture of the GPS up against a beetle-attacked tree. We know that mountain pine beetle is close to the Saskatchewan border. Talking with Rory McIntosh from the province of Saskatchewan last week, so far, at least in northern Saskatchewan, none of the traps have been showing attacks, but that's not to say that they're not there just hiding. So the question is then, will population dynamics of this species differ in these new habitats that are being invaded during this current outbreak? So remember that the historic habitat is down here, but this outbreak has spread well beyond that into areas that we think have never been before colonized by mountain pine beetle. So will the trees in these novel habitats present a different defense, a different host quality to mountain pine beetle, affecting population dynamics? So that's the crux of the work that's going on in my lab. So if we look at, remember I told you that mountain pine beetle can overcome pine defenses because they attack en masse. And so that is density dependent. The more beetles that attack up until a point, the more likely it is that they're going to overcome those defenses and successfully reproduce. But down here where populations of beetles, of attacking beetles, are lower, the defense of the tree can make a difference. So a tree that's relatively undefended will be overcome by mountain pine beetle at relatively low population attack densities, whereas if the tree is well defended, we see that it can fend off those attacks. And so it's thought that this part of the curve, where the numbers of mountain pine beetle are relatively low that are attacking, is affected by both the host genetics and the environment. So this is what really matters if we think about it in an advancing outbreak, because the arriving beetles, those pioneer beetles, are not in huge numbers. So this is where it could make a difference as to how well defended a tree is, what kind of host quality that tree presents at the leading edge of an outbreak. So the first hypothesis that guides the work that we're doing in my lab is that the co-evolved hosts, so lodgepole pine, have different traits of defense and other traits that affect host quality than evolutionarily naive hosts, considering jack pine. So the genetic differences between these guys is contributing to host quality differences. That's our hypothesis. Our second hypothesis is based on a number of ecological studies that have been carried out where they've been able to show empirically that stress trees typically are those that are favored and can be overcome by low-density beetle attacks in the endemic phase, so when beetle populations are relatively low. So stress trees are the favorite target in an endemic phase, whereas healthy trees are favored when those numbers get really high and the beetle can attack in sufficient numbers to overcome a healthy tree's defense, because the healthy tree is posing a much better buffet. So a lot more nutritional quality, much better host quality. So risk-reward, it's worth the risk of trying to attack that tree, because if you win, you get a better buffet. So our hypothesis is that stress trees are going to offer a different host quality, but what is that difference in host quality? And so if we think about looking at climate moisture availability, say using Ted Hogg's climate moisture index, we can see that the historic outbreak typically takes place in a fairly dry region of the country. It's moved across regions that are quite wet. Now it's moving again into drier areas. But we know, particularly those of you who garden, that the last 10 or so years have not been kind in terms of water availability here on the prairies. And so these trees have experienced a substantial amount of drought over the last 10 or 15 years. Has that rendered these trees more susceptible to mountain pine beetle? That's one of our big questions. So this is what I like to call the factorial from hell. So we've had three different factors that we've varied: species, water availability, inoculation treatment, and we've conducted these over a time course, both in growth room studies and in field studies with mature trees. So in each case, we've conducted multiple experiments over time to get at the questions I'm going to show you. So I'm going to show you a bunch of different data to talk about how these trees are responding. And I'm going to interchange between seedling and mature trees. So it'll be marked on the slide whether the data are from seedling or mature tree studies. So pines typically are conservative water users. So when they have low moisture availability, they typically close their stomata to conserve that water. And so that's what we see in our seedling experiments when we apply a water deficit, which is not super extreme, but it's extreme enough that we do see stomatal closure or decrease in stomatal conductance. And we see that photosynthesis also declines because of this reduced gas exchange. So this is in seedlings. We see a similar trend, although not nearly as extreme in our field studies, because we don't see as much of a difference between water availability between these two treatments. But we would assume that the same trend would be there if you could drive as great a water deficit as you can in pods. So a classic stress hormone is abscisic acid. We often see increased levels of abscisic acid, say, in guard cells, when trees are subject or other plants are subject to water deficit. And so we see that the water deficit treatment that we applied to our seedlings does increase ABA levels, but this isn't in leaves, which have been classically studied. This is in the phloem. So the phloem is also responding to this water deficit by increased ABA levels. We don't actually see this dramatic of a difference in the field studies, again, probably because those treatments are less extreme. So one of the measures that's often used to determine not only how is the tree responding to mountain pine beetle, but how the mountain pine beetle would respond to the tree is using the fungus that the beetle carries as a proxy and carrying out inoculations. And so we can measure these lesion lengths, this is commonly done in plant pathology as well, to measure the tree's defense response to that fungal inoculation. But this also reflects the degree of fungal growth. Okay, so it's a bit of a confounded measure, and I'm going to come back to that a little bit later. But what we can see, if we look at seedlings over a time course, we see that under well-watered conditions, we get a response right away. And then that lesion length declines, whereas under water deficit, we see that it takes longer to get to maximum lesion length in lodgepole pine. Same sort of trend in jack pine, although relative to lodgepole pine, that response is a lot more delayed. In mature trees, we kind of have the same pattern as well. We see that lesion length increases more dramatically in our well-watered treatment relative to our water deficit treatment, even though this is a very mild treatment. Whereas in jack pine, we see that maximum lesion development happens later than it does in lodgepole pine. So that's pretty cool. So that fungal growth blocks tracheids in the wood. So that's kind of neat. But this is even neater, that lodgepole pine sees a decrease in stem hydraulic conductance as a result of that inoculation. So because these tracheids are blocked, it's not able to conduct water as easily as it can when it's not been inoculated. But jack pine seems to compensate. So despite the fact that it's got sectors of that stem where conductivity has been blocked, it's able to overcome that, and we see that stem hydraulic conductance isn't altered in jack pine. So here we're seeing some differences in physiological responses that might explain why jack pine can do pretty well in water-limited areas. So we also looked at defense-related hormones. One of the typical ones that's looked at, classic defense hormone, is Jasmonic acid, which is often associated with pathogen responses. And so we see here in our seedling experiments that early and late we see this increase in jasmonic acid levels in inoculated plants relative to wounded plants and controlled plants. Same thing in jack pine. So this is as expected. See something a little bit different with salicylate, which is also a classic plant stress hormone, where jack pine is responding. Lodgepole pine, not so much. But jack pine is really only responding under water deficit conditions. But it's really responding. These levels are really high. So why this is, we're still trying to wrap our head around, but this could represent a different mode of growth of this fungal associate, this Grossmannia clavidra, in jack pine versus lodgepole pine seedlings under water deficit conditions. It's typically we see that a salicylate response in herbaceous plants is often associated with a biotrophic pathogen, sneaky pathogen, versus a necrotrophic or killing pathogen, which is typically the response that we see with this Grossmannia clavidra. So we might be seeing a different response here, or this might be mainly just an effect of the drought. We have to figure that out yet. We've also looked in the mature trees. And so what we see is patterns that are largely similar. We see increases in jasmonic acid under the wounding condition with the pathogen. But our salicylate pattern's a little bit different. Here we see that lodgepole pine accumulates salicylate. Jack pine more so in the well-watered condition. But there's something else that's going on. So that's our local response because it's inside the lesion. It's inside the lesion here. So this is the area where we would think that the fungus is growing. Some initial experiments that Adrian has carried out say that the lesion approximates fungal growth. But we also see some hints of a systemic response, particularly the Jasmonic acid in lodgepole pine, which you see much less of in the jack pine. And then the salicylate in the jack pine, but under well-watered conditions. So that's telling us that the tree is preparing outside of these lesions as well. There are changes going on outside the lesions in advance of the fungus getting there. So that's kind of cool. And you can't get at that so much in seedlings unless you have a death wish and want to try to extract out the lesions. So we've gone on and looked at gene expression, given that these lesions represent this tug of war between the fungal pathogen and the tree. We've gone on and looked at gene expression of genes that are classically associated with defense responses in plants in general and trees in particular. And so what we see are some really neat patterns. So chitinases are a nitrogen-based defense. These guys here are both genes that are involved in terpenoid biosynthesis, and we see different patterns in different members of these gene families. So some of these genes are showing an increase in the constitutive expression. And the underwater deficit. So water deficit is increasing the plant's overall defense posture through increase in expression of some of these defense-associated genes. But we have other genes that, upon inoculation, we see that they're not being upregulated as they are under well-watered conditions. So the induced defenses seem to be compromised. So we have an enhanced constitutive response. So if that's overcome, the tree's got nothing left in the bank to put on as an induced response to try to protect itself. So that's really interesting. But that's only a handful of genes. We have recently been fortunate to undergo genome-wide sequencing of 3 different conifers, and best guess is that they contain 60,000 genes. So here's 4. What about the other guys? So what we've been doing, and Adriana's probably devoted about 30,000 hours to these things, are microarray gene expression analyses where we can look at thousands of genes all at the same time. And so because we're overzealous, we've done a factorial experiment looking at different species under different water availability conditions, under different inoculation conditions, over different time points in 2 different tissues with lots of replicates, which if you're doing the math is a lot of data. So what do we get out of a lot of data? Well, we see that there are thousands of genes that are differentially expressed when you inoculate these seedlings with this fungus. So this is just one little part of the whole story, showing that over time we see that most of the differential expression is happening at 7 days. So we spend a lot of time looking at those 7 days. But lots and lots of genes. So how do you create a story out of all of these different genes being up or down regulated in response to this pathogen? So what we've been doing is a functional approach by looking at the functional categories within metabolism and other cellular processes that these genes belong into. So this is something called MapMan. It was originally created for Arabidopsis, which is a model plant. And so we've been able to use our genes to map onto this schema. And so in this schema, I'm going to show you a few slides just to give you a flavor of what we've been doing. Blue is upregulated in inoculated plants versus control plants, and red is downregulated in inoculated versus control plants. And so each square represents a different gene, and the darker that square is, the more that gene is upregulated or downregulated. So there's lots and lots of different bins in here. The first bin I'm going to start with is this cell wall bin. And so within here, each one of these little sub-bins then talks about genes that are involved in a specific cell wall process. And so we know that there must be a lot of different cell wall changes going on because we can see them using microscopy. So on the left there, we've got control phloem from a mature tree, happens to be a hybrid, versus the next slide, which has been taken from inoculated phloem. So you see not only lots of these phloem phenolic cells that are expanding, enlarging, but you see that it's having a lot of effect on the other cells of the phloem. And so it turns out that within this tissue, lodgepole and jack pine are doing different things with their cell walls. And I just pointed out a few of those differences. So pectinesterases are really involved in those early stages of cell development. So we see that those are really being downregulated in lodgepole pine, not so much in jack pine. Cell wall synthesis activities are greater in lodgepole pine than jack pine, whereas degradation is being downregulated here, upregulated here, modification downregulated in jack pine, upregulated in lodgepole pine. So we can begin to get at differences in what's going on with cell walls in the development of the lesion, despite the fact that we can't see too many differences with microscopy. So we can go on, this is something that Adriana's just done recently, and look at what goes on in the xylem, because we think about the phloem as the primary contact for mountain pine beetle. It has to overcome those defenses, but ultimately the fungus is growing through the xylem, and that's where it's getting a lot of its nutrients. So there has to be a response there as well. So Adriana's been looking at that. This is sort of a generalized schema of what happens after a pathogen starts to invade, right down to synthesis of the secondary metabolites that protect the plant and pathogen-related proteins. So these include those chitinases I was talking about. And I've also shown here, because this is really interesting to Adriana, particularly because of our hormone data, are hormone signaling pathways that might be involved here. And so what I'm showing you here is lodgepole pine over 7 and 28 days under water deficit, well water conditions. And so you see at 7, quite a bit going on. 28 days, still quite a bit going on under well water conditions, but not really under water deficit conditions. And so this is fairly consistent with our lesion length data, where we have a lot more going on early than late. And so we might think that that water deficit conditions, we've probably maxed out the response that we're going to get in terms of the hormone synthesis and hormone degradation, as well as secondary metabolite production. Here we've still got activity going on, so that's kind of neat. But most of this coincides with active lesion development. If we compare that to jack pine, you see that we've still got lots of activity going on at 28 days, and the lesions are still growing at 28 days. So whereas the story is kind of over in terms of lesion development with lodgepole pine at those later days, it's still not over for jack pine. So does that mean that lodgepole pine's kind of given up and the fungus is just going to take over, or did it win? And so that's what we need to find out, and that's what we have to use some new experiments to track where is that fungus relative to that lesion. This is really hot off the press. This is like Monday, where we now have the ability to connect differences in allelic variation for different genes over the landscape with whether or not those genes show a differential response to water deficit, to inoculation. And this is one such gene. We have a bunch of them now. We have about 100 of them, but this is one of them. And this happens to encode a transcription factor that could potentially be involved in defense responses. And you see that if you look at the code here, which is quite small, that has a very low frequency of one allele in jack pine, which goes to a much higher frequency over here in lodgepole pine. So this is a gene that's common across both, showing a strong preference for one allele versus another across this range of hybridization. And so this particular gene is downregulated very late in the response in lodgepole pine in response to Grossmannia clavigerA inoculation, but in jack pine is upregulated really, really, really early and under many different conditions. So here now we have our first hint of a genotype to phenotype correlation where now we can start to get at some of the genetic underpinnings of why are these responses different. So not just the genes are responding differently, but what are the genetic differences that might lead to this difference in control. So with that, I just want to end with a few thoughts. One is on this zone of integration, this hybrid zone. That zone of integration is a lot bigger than we ever gave it credit for. And so we really want to get at that in a little bit more detail in the next few years. Distributions of lodgepole pine and jack pine, that zone of contact I forgot to mention happened relatively recently in evolutionary history. And the distribution of lodgepole and jack pine within that zone of hybridization and the hybrids reflects a habitat specialization, which is probably maintaining that hybrid zone. So lodgepole pine and jack pine hosts show fairly similar responses in their core to this fungal associate when challenged with that fungus, but a lot of differences as well. And those differences may parlay to differences in host quality, which could impact the progression of the outbreak as the beetle moves further east into jack pine. Water deficit affects both the constitutive and induced defense responses. It seems to increase that constitutive response, but if that's overcome, the tree has nothing left to apply to its defenses when we look at a few genes. Now we're looking at this over thousands of genes to see, does that story hold true over the whole ensemble of the defense response? The timing of that defense response also seems to be different under water deficit, and that could be critical if you think about a tree trying to defend itself against marauding beetles. So we've taken the first baby steps to linking genotype with phenotype using genomics, and those are really powerful and they're very promising. At the end of the day, I think we've probably asked more questions at the end of all this than we ever started with now that we know a little bit more about it. And what we really need to know are those differences that we've documented between trees from here and trees from here enough to affect population dynamics? Because that's what really matters when it comes to making decisions about spread control is, will the beetle do better in these trees? If it's going to do better, we need to control more. If it's not going to do as well, maybe we can give it a break. So that's ultimately what we want to do, is try to understand how what we've done so far can answer this question. So with that, I have to thank all the people who do the hard work in the lab, both those that are in the lab now and those who have been in the lab in the past, as well as a whole bunch of collaborators. If anybody knows somebody who wants to also do field work at the same time as doing cool lab work, there's room in the lab for graduate students. And with that, I will take any questions.